lineup of speakers for you this morning. And so to start off this session, let's welcome Guoping Fan from MIT. And he will talk about dissecting synaptic and circuitry mechanisms of autism. Please join me to welcome Guoping.
Thank you, Nancy. First of all, I'd like to thank the organizers, Li Chuan, Nancy, and Carol for organizing such a wonderful meeting. And I also want to congratulate you for organizing such a great meeting and give me the opportunity to present some of our recent work at the here. So my lab is very interested in how the synapses are developed and how their function are modulated. We are particularly interested in how synaptic dysfunction may contribute to neurodevelopment and psychiatric disorders. And this is a drawing of a spine. Many of you are probably very familiar. This is very small, and you have heard a lot of talks about the imaging and the molecular dissection of the function of spines. And so in this, this is about a few microns, 2 or 3 microns in the width, and at the tip of the spine is the postsynaptic density, which is about 200 nanometer dense. But in this very small, tight space, they are, based on proteomic studies, there are probably about more than 1,000 proteins, type of proteins clustered at the postsynaptic site. So it suggests there are many different ways to modulate the synaptic function and control its development and its plasticity. And as we said, that if there are 1,000 ways to build it, there are 1,000 ways to break it. So in the last few years, large-scale genetic studies also suggest that many of the risk genes for autism, schizophrenia, and bipolar, many other disorders are encode genes actually encode proteins at the postsynaptic site, suggesting this is a hot spot for study many of the brain disorders, especially in psychiatric disorders. And the current idea is that the assembly of this large-scale complex is depending on the several scaffolding complexes to assemble, to bring many of the signaling molecules and to the synapse. So one of the major scaffold is the PS95 SAPAP shank complex. So that's one of the main focuses of my lab, try to understand how this scaffolding complex during development help to assemble the postsynaptic density and modulate the function of the synapse. The idea at the postsynaptic side is that this is scaffolding at one end, complex at one end interact with membrane proteins at the postsynaptic site, including NMD receptors directly, indirectly, AMPA receptors, and also with neuroligand urexin, which we have the world expert, Tom is right here, and also potassium channels and other adhesion molecules. And on the other hand, this PS95 family complex interact with SAPAP and shank, and linking to the cytoskeleton or to other signaling molecules, including mGluR signalings and in the complex. And the shank interact with many actin binding proteins or actin modulators, so they can link this this whole postsynaptic complex to the cytoskeleton. One of the major functions is this complex has been proposed is to modulate the trafficking and targeting of this complex to the synapse. So in my lab, we use mouse genetics to dissect the function of these complex in synaptic function and dysfunction. Today I'm going to mostly focus on the shank, one of the shank proteins. We think they play a very important role at the synapse, also relevance to autism. We started this project by looking at the actually SAPAP. I'm going to very briefly go because it's related to what I'm talking about, circuit-specific defect in the Shank3 mice. So there are 4 SAPAPs. They are highly homologous, but one of the SAP, which is SAPAP3, is very uniquely highly expressed in the striatum, which is the only SAP actually highly expressed striatum. And you all know striatum has been is a very important circuits involved in many psychiatric disorders, including OCD, Tourette's, autism, and many other disorders. So the unique expression of SAPAP3 gave us the opportunity to delete this gene and study the function of synapse in the striatum and also look at the behavior. So when we initially deleted this gene, SAPAP3 in mice, what we found is a very peculiar behavior, which is they grooming themselves all the time. So usually you take a mouse out of the cage, what you will see the mouse will running away from us, right? But this is Shank 3 mice, if you can play the video. And you will see that if you take them out, take the mouse out, put it on the cage top. Can you play the video, please? Okay, so you will see it keep grooming itself. Actually, it override its basic instinct running away from us. And so this compulsive like grooming leads to a very severe facial lesion. They're actually grooming their hair and skin off. So that get us really interested to I think this is a model to study repetitive compulsive behavior, and which is there are a lot of studies suggest that this is defect from the striatum. And so we further show that indeed indeed there is a significant defect in the cortical striatal synaptic transmission, both in the AMPA and mediated current. And we also show that if we put the gene back specifically only in the striatum and in the knockout mice, we can completely rescue if you inject the GFP expressing lentivirus into striatum and knockout mice, they all have lesions. But if you inject the GFP tagged SAPAP3 in the striatum, these are knockout mice. But you only express the gene specifically in the striatum, you can rescue the behavior, provide direct evidence that the synaptic defect in striatum actually is the cause of this behavior. So, as you all know, that striatal is very important for many different functions in animals and in humans, and especially cortical striatal synapse is very important for regulating movement. Right? We have Parkinson's disease because of substantia nigra dopamine neuron die, which regulated the cortical striatal synapse function, and we have Huntington's disease, which is a medium spine neuron die. So all these cause very severe motor defect. We are very interested why we disrupt the striatal cortical striatal synapse lead to such peculiar repetitive compulsive behavior. So there are many. This is a very much simplified drawing of a cortical striatal circuit, probably 1,000 times simpler. And for illustrate one particular point, there are many more new studies. You will hear more tomorrow from Bernardo Sabatini about their new findings on new circuits in this basal ganglia to the cortex. But this is just to point that the current understanding is there are two major pathways in the basal ganglia. One is called the direct pathway, one is the indirect pathway. So one of the theory in repetitive compulsive behavior is that maybe these two pathways are imbalanced that lead to recurrent circuits, that overactivation of the circuits lead to repetitive behavior. The idea is if the cortical information come down and activate the direct pathway, this will just inhibit the thalamus, allow the action go through. And if the cortical is mostly activated the indirect pathway, then it will inhibit the thalamus will to prevent the action go through. So the idea is maybe you have a defect in either of the pathways lead to imbalance that lead toward a more active direct pathway action that lead to a repetitive compulsive behavior. So we decided to make a transgenic mouse to test this. What we did was to make a back transgenic mouse so we do not disrupt the expression patterns or splicing. We did not change anything. We just put a flux stop cassette in front of a large back, which was about 250 kilobases. And so the whole SAPAP3 gene is not disrupted. And so this transgenic mouse actually was crossed to SAPAP knockout mouse. Now you have a mouse with a knockout mouse with a transgene does not express. So if you give it a Cree recombinase, then it will Cree out to the stop cassette and then transgene will start to express. So this allows us to cross this mouse into two lines of Korean mice, which is generated by Jing said that Heinz lab at Rockefeller University, which predominantly express either in the D1 medium spine neurons or D2 medium spine neurons. So what we found is if we in the knockout mice, it will re-express SAPAP3 only in the D2 medium spine neurons, actually did not rescue at all. But if we express in the D1 medium spine neurons and predominantly in D1 medium spine, it completely rescued grooming behavior. So one caveat, so that suggests that the defect in the direct pathway that play a more important role in generation of this type of behavior. It also suggests that maybe the imbalance of this pathway indeed may play a role in this repetitive compulsive like behavior. One caveat is that D2 Cree actually is not completely restricted to the striatum. It also expressed in some other parts, mostly in the subgroup of neurons in the cortex. So we actually crossed to a cortical pyramidal cell, EMX1 Cre, and which expresses all the excited neurons in the cortex and the forebrain. And we did not see any rescue of grooming behavior, suggesting that indeed the defect maybe in the striatal direct pathway maybe play a critical role in this type of behavior. So this got us really interested to see whether this type of circuit defect is a common mechanism or is just specific to SAPAP3 gene deletion. So because if it's a common mechanism, because this type of behavior is seen in many different psychiatric disorders, and the cause of these psychiatric disorders are very, very heterogeneous. So we decided to test whether we can demonstrate whether this circuit defect is a direct, a general mechanism for this type of behavior. So there are several ways you can test to test this. One is to look at whether the genes enriched in the striatum and encoding synaptic protein, whether you find enrichment of mutations in human patients. So this kind of project we are collaborating with Broad and right now ongoing. We just finished one batch up, so we don't have the results yet. The second, second approach we did was to try to mutate other genes in the same pathway and highly expressed in the striatum to see whether this is a gene-specific or circuit-specific defect. So we decided to look at shank, which is directly interact with SAPAP, and also there was one shank which was highly expressed in the striatum. So there are 3 shanks. Only shank 3 is highly expressed in striatum. We thought this would be a good test to see whether if you disrupt a different gene, but it's in the same striatal circuit, whether this will generate a similar behavior. So while we were doing this, actually, there were several clinical papers, studies suggesting that, demonstrating that SHANK3 mutation actually is causative to Phelan-McDermid syndrome, which is an autism spectrum disorder. This got us really excited because we all know, besides the language problem, communication problem, one of the major symptoms for autistic patients is repetitive behavior. So when we mutated this gene, what we found is indeed these mice also repetitive grooming their hair and skin off. So provide another layer of evidence that maybe the circuit defect in the striatum, which I'll show you later. Indeed, we found a circuit-specific defect in the striatum that is probably play a critical role in this type of behavior. So in addition to the repetitive behavior, these mice also show 2 very peculiar, interesting behavior. One is social interaction deficit, which has been demonstrated in autism. The other is we call it phobia of novelty. So I'm going to show you just a video. It's much easier to show video to demonstrate this behavior. Can you click the video, please? Thank you. So this is a wild type, this is a mutant. If you put 2 wild type mice together, you will see pretty soon they will find each other, they will start to social, they will basically sniffing and cuddling. And knockout, even you see they bump into each other, when their heads get together, they will separate and they will stay just as far away as possible. This will study chasing, sniffing, and playing. This is a very normal wild type behavior. All the mice do this. And you see they will stay stay far apart and digging and doing repetitive things. So tell us that these mice indeed have a very dramatic social interaction deficit. This is, you see they will play along for a long time. So let's give an opportunity to look at what kind of circuits, what kind of synaptic defect may contribute to this social interaction deficit. The second behavior we found is also very interesting. We call it phobia of novel object. I don't know whether this TV show is available in China. It's called the Big Bang Theory. Basically, it's a whole bunch of MIT graduates and working at Caltech. They are high functional. Some of them are high functional autistic kids. They're really smart, but they have social and deficit, and they have a phobia of changes. They don't like the changes. Right? So these are actually one of the major issues for autistic parents for dealing with we call it insist for the sameness. They don't like any changes. So this, I'm not saying this is identical behavior with humans, but you will see that if you change the living environment for mouse, you will see a very significant difference. Can you click the video, please? So this is not, this time this is a mutant, and this is a wild type. What you can see, we just put a toy, plastic toy in the middle of the cage, and this disrupted their pathway. And you will see wild type mice, they were sniffing and play with it very soon. Get to know, and they're not afraid of. And this you will see that when they're getting close, and they will startle back, and they will never play with it. You will see they're actually startle back, not just not touching. So you will see they are very, very scared, and they will stay as far as away and the corner. And this will squeeze by. They will never try to squeeze by all this. So suggest the new object is really the change of the environment to them is really very they are very scared of this new change and these new toys in cage. So this gives us the opportunity to look at what kind of defect in the mice can contribute to this type of behavior. So as I mentioned, there are 3 shanks, and only one of them, shank 3, is the only one highly expressed in the striatum. We actually did not proteomics. We did a Western blot analysis to see what kind of postsynaptic change happens in these mice. What we found is that there are very dramatic defects in many of the scaffolding proteins, including SAP absolutely reduced, and HOMA, which is linked to mGlu5, I mentioned later, and PS95. They also both NMD and AMPA receptor subunits are reduced in the PSD. So this is consistent with the previous hypothesis that actually shank is master scaffolding for assembly of the postsynaptic site. And we also did Hope cell patch clamp to stimulate the axon into the striatum and recording from dorsolateral striatum, which is most related to the repetitive compulsive behavior. And we found that there are significant reduction of frequency and also reduction of the amplitude, suggesting that there are very significant defect loss, reduction of functional synapses from this, in medium spine neurons. But in the remaining synapses, they also have reduced the function, which is consistent with our biochemical studies, suggesting that, showing that there are reduction of the AMPA and MD receptors. And so we are, so that made us wondering whether there are also imbalance of these pathways in these mice that can explain this type of repetitive compulsive behavior or even social behavior in the future. So we crossed these mice to 2 lines of mice, which are labeling specifically either the D1 medium spine neurons or D2 medium spine neurons. This was from Gene Set and at Heinz lab and characterized by Chip Gerfen at NIMH. This is from Nicole Clark's lab, one of our collaborators at Duke University. And then we can do a whole cell recording to look at the synaptic transmission in these specific pathways. What we found was that to our surprise, actually, we found the defect. We found a significant reduction in both frequency and the amplitude, but it's only in D2 medium spine neurons. We did not find a defect in the D1 medium spine neurons. And this is many EPSCs. If we look at iPSCs, what we found is that also only in the D2 neuron, actually, we have increased inhibition. Only in the frequency, but not in the amplitude. So together, if you look at the iPSC EPSC ratio, you see a significant increase of inhibition in the D2 medium spine neurons in the indirect pathway of these of SHANK3 mutant mice, which is suggesting that in these mice, actually, because of reduced of the excitation, excited synaptic transmission, and increase the inhibited synaptic transmission, the total result is that you have a very much reduced activity of the indirect pathway medium spine neuron. That's leads to the relevant, relatively enhanced excitability of excitability of the D1 medium spine neurons. So this may explain why these mice have repetitive behavior or other potential behaviors. So, as I mentioned earlier, medium SAP3 actually not only shanks not only directly interact with the NMDA, interact with the AMPA receptors, but they also interact with many other. It's a scaffolding protein, so it interacts with many other proteins in the postsynaptic site. One of the 2 of things are very prominently interacted. One is the calcium channel of 1.3, which directly interact with SHANK3 and in the striatum, and the other is through HOMA. It interact with mGlu5. So both of these molecules have been implicated in the striatum, particularly for synaptic plasticity. We want to look whether not only the synaptic transmission, but whether this synaptic plasticity in the SHANK3 mouse are defective, and if it's defective, whether these also have a circuit-specific, pathway-specific defect. So this one of the major plasticity have been well studied by many groups, including one world expert, Robert Malenka, is right here, and several other labs to show that this long-term depression in the striatum is mediated by calcium release and lead to the production of endocannabinoid and reciprocate back to the endocannabinoid receptor in presynaptic terminal inhibited neurotransmitter release lead to long-term depression. So in this paradigm, we actually found that indeed, if we use high frequency stimulation, we can see a very nice long-term depression in the medium spine neuron. But we found a defect in SHANK3 mice only in the D2 neurons, but not in D1 medium spine neurons, suggesting they are also a pathway-specific defect, not only in basic neurotransmission, but also in plasticity. And again, if we look at the mGlu-R5 mediated, if we use DHPG stimulation to look at the mGlu-R5 mediated synaptic depression in the striatum, we also only see in the D2 medium spine neurons, but not in the D1 medium spine neurons. So put together, we think that in the SHANK3 mutant mice, and both in SHANK3 mutant mice, the defect is mostly in the indirect pathway of the basal ganglia circuits that probably contributed to the or caused the repetitive compulsive behavior, and at least most of our recording on the dorsolateral striatum. So we think that maybe for there's a circuitry imbalance in these basal ganglia is maybe a general mechanism for repetitive behavior, repetitive compulsive behavior in not only in the autistic-related animal model, but maybe also in the OCD, Tourette's, and other related neuropsychiatric disorders. So at the end, I would like to thank all the people who did this work. And in my lab, the SAPAP3 work was initial work was by a graduate student, former graduate student Jeff Welch, followed by postdoc Jing Lu. The SHANK3 initially was done by two postdoc former graduate students Juan Peck and Katia Feliciano, and a lot of physiology. The new pathway-specific physiology in SHANK3 was done by two postdoc fellows, Wen Tingwan and Zhang Yanfu, and several other people in the lab helped. And we have a lot of great collaborators, and without them, this will be will be impossible. And thank you.
Thank you, Guoping, for a very inspiring talk. Yes.
Hello here. For the behaviors, I want to ask whether these mouse feel pain or itch so that they keep grooming.
Yeah, so we did a basic pain study. They feel pain. And so if you look at it very carefully, we cannot tell we cannot tell whether they feel itch or not. But if you look at the behavior, if you show the video, itching usually they use a hind limb to actually scratch them, and grooming, especially facial area, they actually very protective. They actually use this side of so from this kind of view, you can see they are not actually scratching. They are actually grooming themselves.
I think models are sometimes in the eyes of the beholder. So if I was a researcher on atopic eczema, I would say this is a great model for atopic eczema. It's a disease of the skin. It may have psychological underpinnings as well as genetic underpinnings, and people just scratch incessantly. It's hard to be sure that they're not scratching from where I was sitting, but I'm wondering why you leap to autism as the disorder that you're modeling as opposed to a dermatological condition. It's
a very good question. So for the SAPAP, for example, we re-express only in the striatum. We can eliminate this behavior. We did not change the skin. It's still full knockout. All the skin cells are knockout of SAPAP.
But if there's a neuropsychological component to eczema, presumably that's taking place in the brain, and then there's some impact on people's perception of something in the skin. I'm not an expert in this.
So we actually
alternative disorder that this could model.
Right, so it's a very good question. We did a lot of study at SAPAP3 to look at we cannot tell we sense anything, but we crossed many different mouse lines to look at the sensory neuron development, sensory innervation nerve terminals. We did not see any, and the skin inflammation, all these things. So I cannot say there's no central component lead to a sensation in the skin that lead to this kind of grooming.
I don't know autism, but do autism patients have that degree of skin?
I don't think that's a major issue, especially for SHANK3 mutation. SHANK3 is known SHANK3 deletion actually cause Phelan-McDermott syndrome, and I don't think that any of them have this type of skin problems.
So I have one question. Of expression, the SAPAP3 proteins only in the overexpression in the T1 neurons can rescue the phenotype. But in the SHANK3 mutant, the phenotype is caused mainly by the D2 neurons. So do you think these two proteins influence in the same pathway or separate pathway?
Yeah, it's a very good question. I don't have an answer now. I think what happened, that's what I think. I don't have proof for that. I think from many of the studies, including Tom Sudoff's study, show that a single mutation can cause a variety of different defects. And from many other studies, you can see from Fragile X syndrome, Rett syndrome, the defects come from all over the place and can affect the synapse very different way. So what I think is that at the end, what happened to the basal ganglion circuits is depend on what is the signal coming back from the thalamus to the cortex that drive the behavior. So I think we don't understand it at this level. Maybe the real converging mechanism, if you want to group many of the things together, maybe at the network level, going back to the circuits. Maybe whether it's D1 defect or D2 defect, they all come to a similar way to affect the network activity in the cortical area. So that's what we are studying right now.
So have you checked, for example, the SHANK3 expression level in the SEPAP3 mutant mice?
We have not checked, but people have done other mutations in SHANK2. For example, if you knock out SHANK2, SHANK3 is dramatically increased. But as I showed in SHANK3, the SEPAP3 is not increased. As I showed, it's actually reduced.
Okay. So the deficits in the D2 indirect pathway explains well the OCD-like behavior cannot stop the ongoing activity. But how do you think that could lead to autism-like behaviors, like unsocial?
So, we don't know, but my personal view is that a similar synaptic defect probably happened in many brain areas. I didn't show you we showed that if we look at the hippocampus in these mice, actually there is no obvious synaptic transmission defect, but it does not mean they doesn't have a detailed, subtle plasticity change. So what I think, the repetitive behavior and the social behavior, the difference may reside on affect different brain regions. And so, for example, one of the main theory is one of the theory for social behavior is the social rewarding behavior of the autism. So we are testing that. So what I think is coming from different brain regions instead of different synaptic mechanisms.
Thank you.
I have a question about the D1, D2 specificity of these two different scaffolding proteins. Can't remember, did you say that, are they differentially expressed? What's the basis for one affecting D1? Yeah,
so we don't know exactly. So what we know is SHANK3 and SEPAP3 are expressed in both D1 and D2. We don't know whether there's a level difference. It's very hard to study because the protein all goes to the spine, so we cannot say how much in the cell body. But you probably remember Ned Hines' lab with Mary Hyman now at MIT. They did a backtrap experiment to specifically look at a gene expression in D1 neuron, medium spinal, and D2 medium spinal. Although these two groups of neurons are very similar in many sense, but their signaling pathway are very, very different. So in theory, you don't need a differential expression. If this downstream signaling is coupled differently from these proteins or bring to the synapse differently, you will affect different pathways. You have a completely different defect. That's what our guess is.
Do you think the direct and indirect pathways are simply parallel pathways or they're crosstalks?
I'm sorry?
Between the two? The direct and indirect pathways that you presented are parallel pathways or they're crosstalks between the two?
Very good question. I think they are I think they are both parallel pathways, but they also crosstalks. And there are people studying whether they are also actually D1, D2 neurons or collateral interactions, and they do have some. And it's still controversial. Some people, we actually did some experiment. We didn't see a lot, but other people reported there are a lot of collateral interactions. So they probably, my guess is, the thing I didn't mention is there are many called microcircuits in the striatum. They are cholinergic neurons, GABAergic neurons. They all regulate the medium spinal function. And some subgroup of interneurons actually differential regulated D2 and D1 neurons. So there are many microcircuit regulations of differential regulation in these two pathways.
Do you have what's the reason that the grooming deficits, one in the SAPA seems like is in the face, the other one seems like on the back?
Very good question. So we don't know exactly why, but there are two group of mice, many of them, not only ours, like Slick Track 5, the phenotype is identical to the SAP episode. The only grooming head, but they are HOXB8. They're extruding almost like Shank3. They're grooming the whole body. So grooming it's called a syntax behavior. They have precise where to start and continue and finish. People have done lesion experiment. If you cut off the cerebellum, you cut off the hippocampus, the rats or mice still will grooming syntaxically. They do the same thing. But if you damage the striatum, they would disrupt their syntax. So one of the ideas is that in the SAPAP3 mice, they couldn't finish. They don't have the feeling that they have finished this part can continue. And so they can go back again and again. So that's kind of...
do you have any motor deficit for the...
So roughly, if you do rotor rod, they are all normal. We did that.
Let's thank Guoping again for a very good talk. So the next speaker is also from MIT, Li Hui Cai, and she will talk about chromatin remodeling in regulating gene expression and cognitive performance. Let's welcome Li Hui.
Thank you. So I would like to open my talk by thanking Nancy and Lichen and Carol for inviting me. So I think it is increasingly clear that epigenetic mechanisms hold enormous power in shaping the complicated gene expression program in the nervous system. And to us, particularly intriguing is how neurons rapidly turn on gene expression following activity stimulation. So just a little bit about the mechanisms. It includes DNA methylations, which is chemical modifications of the cytosine residue, and covalent modifications of the histone proteins, among several other mechanisms. And the modifications of the histone proteins have a particular impact on the overall configuration of the chromatin, particularly histone acetylation. So the acetylation moiety is negatively charged. So increased acetylation potentially, of the histone protein potentially can propel protein-protein and protein-DNA interactions, and therefore force a more relaxed or open state of the chromatin, which supports gene expression because it permits the access for transcription machinery to activate the genes. Conversely, histone deacetylases, or the HDACs, remove the acetyl moiety from the histone tails, and therefore facilitates the more inactive, compact state of the chromatin. So concerning the relationship of gene expression and the function of the nervous system, I think it's the best elucidated in the hippocampal circuits. So I think it's fairly well established that following novelty or other sensory stimuli, there's activation of network ensembles. And this in turn triggers specific gene expression program, which is known to be necessary for long-lasting synaptic modifications and eventually memory consolidation. And the importance of the gene expression, I think, has been demonstrated many times through loss of function experiments, for instance, knocking out or loss of function of certain key transcription factors can have detrimental effects on synaptic plasticity and various behavioral performances. But how about increased gene expression? What's the consequence of that? And it turned out that it is fairly easy to increase gene expression through the epigenetic mechanism. So the histone deacetylases, as I said, that they normally promotes the inactive state of the chromatin. So small molecule chemical inhibitors of the HDACs then can promote the chromatin toward the other state, the more active state, and facilitate gene expression. So, such experiments were done in the nervous system and actually were published almost a decade ago by many groups of investigators. And overall, they observed a very beneficial effect in synaptic plasticity. So, here, for instance, David Sweat's group showed that treating animals with HDAC inhibitors can facilitate spatial and associative learning and enhance synaptic plasticity in wild-type mice. And then other groups have shown that this is beneficial even in mutant mouse strains normally impaired in LTP learning. So, so Andrew Fisher, a former postdoctoral fellow, encouraged by these studies, so he wanted to know if increased gene expression program can be beneficial for memory impairment following neurodegeneration. So he used the inducible P25 mouse model, which can manifest profound neuronal loss and synaptic loss, as you can see here in the hippocampus CA1 area. But after he treated these animals with HDAC inhibitor, he found that synaptic density can be readily rescued. And this is associated with recovered learning and memory performances. So after this, Jisang Guan, another former postdoctoral fellow who is now a PI at Tsinghua University and who is at the conference, he subsequently identified HDAC2 or histone deacetylase 2 to play a major role in regulating the expression of neuronal genes supporting learning and memory. So he found that HDAC2 loss-of-function mice have increased synaptic density and facilitated Scheffer collateral CA1 long-term potentiation. Conversely, HDAC2 gain-of-function animals have reduced synaptic density and impaired hippocampal LTP. And Jisan further performed biochemical experiments, including chromatin immunoprecipitation, to show that HDAC2 is recruited to the promoter of a plethora of neuronal genes known to support memory formation, such as activity-regulated genes and genes involved in synaptic remodeling, including Shanks, you just heard, and genes directly involved in synaptic plasticity. So there, HDAC2 negatively influences their expression, but following activity stimulation or novelty exposure, HDAC2 becomes S-nitrosylated, and this triggers the dissociation of HDAC2 from the chromatin, and therefore results in a cascade of events leading to the formation of the more active state of the chromatin and supporting gene expression. And most recently, Johanna's graph in the lab found that HDAC2 is susceptible to neurotoxicity regulation as well. In this case, neurotoxicity treatments such as oligomeric A-beta peptides can increase HDAC2 expression. And the increased HDAC2 expression results in an increased association of HDAC2 with its target genes, and this in turn results in the chromatin to be locked in the inactive state. And the chromatin is resistant to activity stimulation induced gene expression. And eventually, this leads to cognitive disability. And we think that this actually plays a very important role in cognitive impairment in neurodegeneration conditions. And one line of evidence is that when Johannes looked at HDAC2 levels in a large number of post-mortem human Alzheimer's disease brains, he found that nuclear HDAC2 level is markedly increased. And recently, there are also many other groups found that HDAC2 can be upregulated by other type of toxicity, including behavior stress. So it seems that HDAC2 elevation can be broadly involved in various types of memory disabilities. So I think these bodies of work nicely demonstrate the importance of chromatin remodeling in regulating gene expression in the nervous system. But how about DNA methylation, which is actually the first discovered epigenetic mechanisms in regulating gene expression? So DNA methylation occurred on cytosine, especially the fifth carbon of cytosine. And it predominantly happens on the CPG dinucleotide. And in general, DNA methylation at the promoter regions typically leads to transcriptional silencing. And in fact, majority of imprinted genes are hypermethylated. So methylation is carried out by a group of enzymes known as DNA methyltransferases, or DNMTs. So if there is a mechanism to methylate DNA, there's got to be a mechanism to demethylate DNA as well. So DNA demethylation is believed to cause transcriptional derepression. But to date, the mechanisms behind demethylation are still rather unclear. So there is known to have passive demethylation of DNA, but there are also active demethylation. And the active demethylation is speculated to undergo some intermediate stages before the DNA becomes fully demethylated. And one of the intermediate stage turned out to be hydroxymethylation of DNA. So all of these observations were made fairly recently. In 2009, Angina Rao's group identified the 1011 translocation protein family known as Tet proteins. And she found that the Tet proteins is capable of converting 5-methylcytosine to 5-hydroxymethylcytosine in mammalian DNA. So the Tet family proteins contain 3 members, Tet1, 2, and 3, and they share fairly conserved sequence homology. Also, Nehein's group, he first demonstrated the existence of hydroxymethylated DNA in the mammalian brain. So here he showed, using 2-dimensional thin layer chromatography, that he found, he called it spot X, which is more enriched in the Purkinje cells compared to the granular cells in the cerebellum. And he was able to eventually demonstrate that spot X is actually hydroxymethylated DNA. More recently, Hongjun Sang's group showed that hydroxymethylation of 5-methylcytosine by Tet1 promotes active DNA demethylation in the adult brain. So the question now is, what is the functional consequences of Tet1 promotion of DNA methylation? To address that question, we collaborated with Rudolf Jenisch at the Whitehead Institute. Rudolf generated Tet1 deficient mice by deleting exon 4 of the mouse Tet1 gene, and this resulted in a complete ablation of Tet1 expression in these animals. However, these animals appear to be extremely normal. They are completely viable, seems to be very healthy, and live to an old age. So we subsequently look at their brain, and we couldn't detect any differences in terms of total neuronal count or distribution of neurons between the control and the knockout animals. So we subsequently looked to see the global level of methylated cytosine versus hydroxymethylated cytosine in the brain, in the cortex or in the hippocampus. And it turned out that Tet1 deletion leads to very minor differences in the levels of 5-methyl-C or 5-HMC. So there is very minor increase in the total content of methylated cytosine, and a small reduction in 5-hydroxymethylated cytosine. And the reduction is probably perhaps more pronounced in the hippocampus. So with these differences in DNA methylation and hydroxymethylation, is there a consequence in gene expression profiles? So we perform microarray gene profiling analysis of the control and Tet1 knockout, looking at the cortical and hippocampal tissues. And it turned out that Tet1 deletion really has very minor consequences in gene expression. Altogether, only about 200 genes show significant differences in expression profile, considering there are at least 30,000 transcripts being expressed in the brain. And if you look at a scatter plot, especially in the hippocampus, you can see that most of the genes that show differential expression are downregulated as opposed to upregulated. And the genes within that group include synaptic proteins and ion channels. But really, a small subset of genes that really stand out to us are these genes that are known to be activity-induced immediate early genes, such as ARC, EGR proteins, Npas4, and CFAS. So we further perform quantitative PCR to look at the expression levels of these genes in the naive mice, and especially in the hippocampus. I think you can appreciate that the expression of all of these genes is pretty much diminished. So among these genes, we're particularly intrigued by Npas4, because a couple of years ago, Yingji Lin, a young investigator at the McGovern Institute, she found that Npas4 is readily induced by depolarization in hippocampal neurons. But more interestingly, in the Npas4 knockout neurons, the expression of other immediate early genes is abolished, even following depolarization treatment. So it seems that Npas4 is upstream of other immediate early gene expression. So we decided to look at the methylation of Npas4 promoter by doing sodium bisulfite DNA sequencing. So, so especially we found that in the promoter exon 1 junction of Npas4 locus, it is highly enriched with the CpG dinucleotides. Altogether, there are about 40 of them in the immediate promoter and exon 1. So if you compare DNA methylation content in the control versus knockout in the cortical and hippocampal tissue, I think you can immediately appreciate that there is a huge elevation of DNA methylation in the Tet1 knockout. So in the cortex, this is, normally in the wild-type animal, there's hardly any methylation, 4%. But in the knockout, about 24%. In the hippocampus, the difference is even more striking, 58% of the CpG islands are methylated. And this is in stark contrast to what I showed you earlier, the global levels of DNA methylation, which only show a very, very minor reduction. So this suggests that Tet1 really targets a small subset of genes in the nervous system. So we decided to then look at whether there's any behavioral consequences with such a reduction of immediate early gene expression. And completely to our surprise, turns out that these animals look extremely normal, okay? So here I'm just showing you a couple of results from using the hippocampus-dependent learning paradigms, such as Pavlovian fear conditioning, to look at associative learning, and we really do not see any differences between the two groups of mice, either in the contextual learning or Q learning. Similarly, the Morris-Water maze evaluating spatial memory acquisition, again, the two groups of mice just simply don't show significant differences. So this is surprising, given that there is a true differences in gene expression. So we said that perhaps we should look at the expression of these immediate early genes again after behavior training. So here we go. So we harvested the tissue from these animals after the CSU was pairing, and 60 minutes later, we look at this immediate early gene expression, and it turned out that they no longer show differences in expression, both in the cortical tissue and in the hippocampal tissue. So we wanted to really see, whether there's any differences that we can detect in terms of the two groups of mice, in terms of their behavior. So we've subjected them to a large battery of different behavior tests. And we finally found that there is one difference that we can tell, which is turned out to be memory extinction. Okay, so using the fear conditioning paradigm, there is a very well-established protocol to extinguish the fear memory, which is after the animals acquire the fear memory, then if you re-expose the animal to the same context and for a long period of time, allowing the animal to explore, then eventually the animals probably will decide that this context is actually safe rather than fearful. And therefore, their fear memory level would decrease as expressed by reduced freezing levels. And this is exactly what we observed in the control group of animals. But in the Tet1 knockout, we simply do not see any difference in terms of the freezing level. And this result was further supported by the spatial learning extinction as well. So here, after the animals acquired the spatial memory, we just remove the hidden platform from its usual place. And then we put the animals back to the swimming arena and then measure the time spent in the target quadrant where the platform used to be, and also the number of platform crossings, although the platform was no longer there. And then we follow the two groups of mice. And in the first 2 days, we didn't observe a difference or a significant difference between the two groups. But by the third day, I think you can appreciate that now there's a huge difference in that the control animals continue to decrease its time spent in the quadrant or crosses, but the knockout animals do not show any reduction at all. So we also look at electrophysiological correlates of these observations. And we took acute hippocampal slices and look at basal synaptic transmission, pair pulse facilitation, and again, they look very normal in the knockout. And the Sheffield collateral CA1 LTP after theta burst stimulation, also there's no difference between the two groups. And the only difference that we can see is actually after low frequency stimulation. We found that the Tet1 deficient mice seem to have facilitated long-term depression. And so far, this is the only electrophysiological phenotype we can observe. Finally, we look at immediate early gene expression again, this time following extinction training. So, so here, to our surprise, we see a difference again. So look at Npas4 and CFOS. There is a reduction in the Tet1 knockout groups compared to the control group. And ARC, there's also a trend in reduced expression following extinction training. Finally, we look at, again, methylation of the Npas4 promoter by sodium bisulfite DNA sequencing. And here, you can see again, in the control animals, there's very little methylation of DNA in the cortex and the hippocampus. But in the knockout, we see increased methylation of DNA. Although if you look at the extent of methylation, it's not as high as in the naive mice, but still there is a significant difference between the two groups. So, so this is a summary. We think that this data suggests that Tet1, although is not required for expression of many, many genes in the nervous system, it is critical for the expression of a subset of neuronal genes, especially activity-regulated genes. And we think that the physiological role of Tet1 is to prevent a hypermethylation of promoter areas of its target genes, and therefore maintaining the plasticity of the chromatin capable of responding to stimulation. However, based on the differential methylation levels and gene expression levels following different behavioral manipulation, we speculate that following a strong stimulation such as fear conditioning training, Tet1 deficiency can be overridden perhaps through the compensation of the presence of other family members, such as Tet2 and 3. Both are highly expressed in the nervous system. However, weak stimulation, such as extinction training, fails to completely elicit the compensatory mechanisms. So finally, this is a schematic really to bring home what we think is the function of Tet1 at the chromatin. So in the absence of Tet1, we think that the DNA of its target gene is hypermethylated and therefore is locked in a very inactive state. But Tet1 can cause hydroxymethylation of these sites and perhaps eventually leads to demethylation of the DNA in the promoter. And this renders the chromatin more plastic and now can respond to activity stimulation, induced dissociation of the co-repressor complexes from the chromatin, and therefore acetylation, relaxation, and permits the access for transcription machinery to turn on gene expression. So with this, I would like to thank the people who have done the work, Andre Rudengo, a postdoctoral fellow in the lab, and Milad Daulati, who is a postdoc in Rudolf Janisch's laboratory. And I would also like to thank my postdoc, Johannes Graf, and my former trainees, Andre, Jisan, and Jun. And I would like to thank my collaborators and the funding agencies. And thank you very much.
We have time for a few questions.
May I have a quick question? Have you tested the reversal learning for the tech knockout mice?
Yeah, I think it's a very good question. We are in the middle of doing that. Yes.
At the beginning, you showed a picture about the HDAC2 KO mouse, and its HDAC2 affects the memory of the mouse. And at what stage do you do the experiment? Because I remember Eric Olsen said HDAC2 knockout leads to perinatal death of the mouse.
So that's not our impression. So the HDAC2 knockout we used is actually a whole body knockout, and they are actually completely viable. And but we have also used the HDAC2 conditional knockout in the KIMK2 positive neurons, which show a very similar phenotype.
Okay. And in the last model you showed, I remember on Npas4, you showed the gene structure is around the gene's transcriptional start site, right? In the last model, you showed the HDAC2 binding to the maybe interact or colocalize with the Tet1, maybe also located on the gene transcription start site. But as the transcription start site, HDAC histone demethylase not usually inhibit gene expression because some histone modification, the methylation state is required for the gene expression.
Yeah, of course. I mean, I really just show a very reduced picture of the complexes sitting on the promoter region. There are a lot more components there. And in terms of possible interaction of Tet1 with histone deacetylases, Christian Helin already published a paper showing the presence of the Tet proteins in the HDAC complex.
Okay, thanks.
In the back.
I have two questions. One, is any of the genes that interact with Tet1 imprinted? And if so, is any difference between the male and the female behavior?
Yeah, so far we, at least from our microarray analysis, we really don't see imprinted genes that show differences in expression levels. But I think it really, this type of question really needs to be addressed by a genome-wide, perhaps chromatin immunoprecipitation deep sequencing analysis to be addressed. And I know that there are people who are doing that right now.
The other question is that, as you know, some of the gene expression is mostly the active versus the inactive would be related to the location in the nuclear matrix. Have you shown any of these hyperactive genes to be in the achromatic area of the chromosome, or sorry, of the nucleus, or the heterochromatin and the inactive ones?
So what do you mean by hyperactive genes?
When you see, you have two sets of genes that overexpressed and other are underexpressed. So those will be in a different location.
Oh, you mean those genes that show
the nuclear position?
Yeah, that's a good question. We haven't looked at that yet, but eventually that needs to be done.
Liwei, I didn't hear the ages of the mice. I assume they were adolescent, but have you considered looking at the effects of aging on your...
You mean the Tet1 KO?
Yeah, the Tet1.
So we've done most of the experiments in 3 to 4 months old animals.
Would it be worthwhile to see if older mice, 12
Yeah.
months, 20 mice?
Yeah, great suggestion.
Yeah, okay.
Yeah. Thank you.
Yeah. I have one question. A lot of studies have suggested extinction is also a kind of learning. So, but in the mutant mice, only the extinction was impaired.
Right.
So, do you think the difference is only due to the stimulation intensity you suggest, or due to another the differences between these two kinds of learning? For example, in hippocampus, in the mutant mice, the LTD is impaired. So
Yeah, that's a very good question. Obviously, it has been suggested that the extinction learning is a weaker paradigm compared to, say, memory acquisition type of protocol. And also, interestingly, in the literature, I mean, obviously, I really don't know the precise mechanism, but memory extinction phenotypes very often associated with LTD differences as opposed to LTP.
Okay, if there are no more questions, please join me to thank Liwei again for an excellent talk. Okay, the next speaker is from UPenn, Virginia Lee, and she would talk about her work on transmission of misfolded proteins in neurodegenerative diseases. Let's welcome Virginia.
As you can see, I've changed the title of my talk, and I realized that in 20 minutes I can't possibly talk about all the different neurodegenerative diseases. So I restrict my talk only to alpha-synuclein transmission in Parkinson's disease. But before I do that, I want to thank Nancy and the organizer for this lovely meeting in Costa Spring Harbor, Asia. So many of you are familiar with these common neurodegenerative diseases. They're characterized by the accumulation of misfolded disease proteins, and you're quite familiar with Alzheimer's disease and Parkinson's disease, where senile plaques comprised of the A-beta peptide and neurofibrillary tangle comprised of tau protein accumulates. And in Parkinson's disease, those inclusion bodies are called Lewy bodies, and they're comprised of the protein called alpha-synuclein. And so my lab has been interested in trying to understand the pathophysiology and biology of tau, alpha-synuclein, and also TDP-4A3 and other accumulation and in ALS and FTD. But we wanted to develop models to study these diseases in both in animal and also in cell. But we really, have to learn from other fields in the sense of trying to understand how these misfolded proteins accumulate. So we turned to the prion field because they actually have studied the pathogenesis a little bit earlier than the rest of these different neurodegenerative diseases. And so, what are prions disease? And many of you know that prions disease are infectious, and the common infectious neurodegenerative disease of prions are mad cow disease or CJD. And so, and many of you might remember, 30 years ago, that when Stan Prusner came out that, these prions disease are not comprised of prion particles that are responsible for the infectivity, are not comprised of DNA or RNA, but instead comprised of only protein. And so, based on this hypothesis proposed by Stan or others, and others as well, that normally the prion protein, which is called PRPC, has a normal conformation, but it can also adopt a pathological misfolded conformation. And these PRPSC, the pathological form, can aggregate very slowly to form these seeds. And then once they form small seeds, they also recruit monomeric normal PRPC and to form larger and particles, and then eventually form larger amyloid seeds. But little bits of these fragments can also be, or seeds, can also be broken from the larger seed and start the cycle again. And even though that this is very compelling because it means that a normal protein can, a normal protein can template also corrupt the normal conformation to form amyloid. But as you know, that neurodegenerative diseases are not infectious, so basically, we think that neurodegenerative diseases may be transmissible in the sense of spreading in the brain, but they are not infectious like prion disease because there's no evidence to support that Alzheimer's disease and Parkinson's disease are infectious that can be transferred from man to man. And so what are the evidence to demonstrate that, perhaps that this spreading phenomenon occur in Parkinson's disease? And so the first piece of evidence and came from the work of Heiko Brack about 10 years ago now, and where he basically stained a large number of brains with Parkinson's disease and then basically categorized them in terms of the abundance of the pathology and the distribution as well. And so basically he always observed a stereotypical progression of pathology. They always start in the lower brainstem, in the motor neuron and nuclei of the vagus DMV. And then they move upwards and to the upper midbrain, to the brainstem, upper brainstem to the midbrain, and then eventually spread throughout the brain. And so there's one evidence that at least in brain specimens, that you can see this progression. And the other piece of data that came out about 5 years ago is very, very provocative in the sense that, as some of you may be aware, that in the 90s, in the 80s and 90s, one way to treat Parkinson's disease patient is to transplant fetal mesocephalic graft into the patient's brain. And some of these patients that have this transplant and eventually die 10, 12 years later, and then the brains were examined by a neuropathologist. So two different groups, the group of Jeff Cordova and also Patrick Bunden, came up with the same observation in the sense that they were able to detect Lewy bodies that are comprised of alpha-synuclein in the graft of these patients that had the transplant and many, many years ago. So supporting that perhaps that there may be something that the bad cells that have the pathology can spread to the good young cells and then corrupt them and so that they would develop pathology. Making alpha-synuclein aggregate in cells and modeling alpha-synuclein aggregation is really very challenging. And part of the reason is that alpha-synuclein is extremely soluble protein, and overexpression alone does not lead to disease-like inclusion that you can see here. And we reason that perhaps then we can develop seeded alpha-synuclein pathology in cells and in also animal models. So how do we do that? Well, despite the fact that we can make these protein aggregate in cells, you can readily make them aggregate in a test tube. So you can take recombinant protein and then you basically, and manipulate them under artificial conditions, and you can see that they aggregate readily to form these amyloid fibers. They're very similar to what you see in the human brain. And so here, basically, I just show you that wild-type protein aggregates a bit slower than the mutant protein, the A53T mutant protein, and which is one of the major mutations found in familial Parkinson's disease. But the other observation that's really intriguing is that you can add small amount of these, what we call preformed fibers, or PFFs. And you can add a small amount of this to a monomeric solution, and you can reduce its lag time. And so in other words, that the kinetics of fibrillation become very, very rapid. So then we asked that, can we take advantage of this assembly properties and to generate Lewy body-like inclusions in neurons? So basically what we did was that we took preparations such as this, and then we sonicated them, and we just throw them into primary neurons that are not overexpressing anything. They're just mouse neurons. And so this is what we saw 14 days later. And using an antibody to phosphoalpha-synuclein, which is marking pathology. So normally synuclein is not phosphorylated. Only when they develop pathology and they acquire this phosphoepitope. And so you can see the pathology is really very, very extensive and really quite impressive. And these aggregates are insoluble as well. And so how did they start? So initially, when you throw the fiber in, 4 days later, you start seeing the pathology at the presynaptic terminals, and that's where alpha-synuclein has the highest concentration. And when you wait for another 10 days or 14 days later, you can see now the pathology spread to the entire cell, to the cell body and dendrites and so on. And you can see that they're indeed comprised of amyloid fibrils because you can label these bundles of fibrils with an antibody to phosphosynuclein. We can actually even demonstrate that these little bits of fibrils are actually internalized by neuron, and you can see using this confocal images, but other methods that we've used to demonstrate that indeed these little bits of fiber can go inside and can be transported. And so there are these, we characterize the pathology, and they're very similar to those that you find in human, and both morphologically and biophysically, and also they acquire the same biochemical modification, such as ubiquitination and phosphorylation. And having the pathology inside the cell and really compromise normal cellular function. And this is one example on synchronization. You can see that the synchronization is compromised in the PFF-treated culture compared to the PBS or non-treated culture. And you can see that over time, the neurons do die. So having the pathology is not good for the cells. They eventually do die and over time. And so what I've told you so far is that we can generate this pathology in cells, but I haven't demonstrated to you they spread. And so to really demonstrate a cell-to-cell transmission, and we use this microfluidic device that comprised of 3 different chambers. So in this device, what you do is that you put cells in chamber 1, 2, and 3. And then the only way they can talk to each other is through these grooves, and so they allow the axon to go through. And so we reasoned that if you put fibrils in chamber number 1, and then now over time you observe pathology in chamber number 3, that is pretty reasonable, good evidence that transmission and cell-to-cell transmission occurs. So indeed, this is what we saw. And you see that after 18 days, and we put the fibrils in chamber number 1, you see pathology going through the grooves into chamber number 2, and then you can see the pathology going through the grooves from chamber number 2 to chamber number 3. So what I've shown you, and this is part of my talk, is that the misfolded alpha-synuclein protein is sufficient by itself to seed the recruitment of endogenous protein into insoluble deposits. And that endogenous alpha-synuclein is converted and accumulate as inclusion that share key features of Parkinson's disease pathology. And this small quantity of exogenous material and can initiate pathological cascade leading to neuronal dysfunction and reduced survival. And that we show that cell-to-cell transmission is responsible for the spreading of this pathology. So the big question is that how does this relate to what is observed in human alpha-synuclein, human synucleinopathies? So to do that, we first turned to transgenic mice that overexpress alpha-synuclein. So a while ago, we published this mouse model where we overexpressed the mutant A53T human alpha-synuclein in mice. And by about 8 to 16 months or so, these mice die. So we argue that, if we can then take young mice, the same transgenic mice, and then we take some of the mice with the pathology, and then we make a brain lysate, and we inject it into these recipient mice. Then we ask, can we then demonstrate that this misfolded alpha-synuclein elicit conversion and accelerate aggregation? In other words, that whether we can observe seeding phenomenon in this recipient mice, and then also whether the seeding can propagate along and to neighboring cells, in other words, spreading. So we injected in the striatum and the cortex, and 3 months later, and we saw really some quite spectacular pathology. And so these mice are only about 5 months of age, and you can see the pathology spread all over the brain, the cortex, brainstem, everywhere. And so obviously, with this experiment, we don't really know, because we inject brain lysate, we don't know whether or not alpha-synuclein itself is implicated. So to do that, we basically inject alpha-synuclein fibrils directly into the brain and ask whether or not, and we see the same thing. And so we use two different synthetic and alpha-synuclein preformed fibrils. One that is truncated, and then we can tack it with a MIG-tack, and also we can follow it because we can actually use antibody to the C terminus and to attract the recruitment of the endogenous gene. So basically, we saw after even 30 days and post-injection, and you see pathology already in many different regions, and definitely in the injection site. But by 90 days, the pathology is really very extensive. So both the truncated and also the full length look exactly the same. And the pathology is much more abundant than the symptomatic mice. So remember I told you that these mice do get disease and they die. And the pathology is really modest compared to the injected mice. And so suggesting that the signal seemed to be amplified. And we mapped the distribution of the pathology, and basically the 2 points I want to make here, and that both of the lysate and the fibro-injected mice show similar temporal spatial distribution of phosphosynuclein pathology. And so suggesting that they are, following a certain specific path or connection and that account for the spread. And the other really surprising finding is that these mice die prematurely. And so normally these mice has an average lifespan of about maybe a year or older. But then when you inject it with fibrils, and then you can see they have a shortened lifespan. And these mice, when they develop the shortened lifespan, they actually show recapitulate the same clinical phenotype of motor impairment and before they die. But what really is interesting is that you see here that we inject the mice over a period of 3 months, or from 2 to 5 months. And you can see it doesn't matter when you inject, they always die about exactly the same time, between 100 to 125 days, suggesting that there is some sort of process that the propagation, that the program of propagation that the mice follow. And also we took a hemizygous mice, and then we injected them with fibrils, and they survive a little bit longer, suggesting there's this dose effect here. So in other words, if you have more synuclein, the mice die sooner. You have less synuclein, and you attenuate this window of survival. So how did it spread? I mean, it spread so quickly throughout the brain. How does it do it? We think that they spread by the white matter tracks. You can see here in this sagittal section, the pathology is really very impressive. You see it all over the place, and including even in the cerebellum. So suggesting that perhaps there are transsynaptic transfer of the pathology occurring. And in the white matter, you can see it in the corpus callosum here, and also in the cerebellar white matter. And also this type of phenomenon can be demonstrated using and brain lysate from human PD brain. So here we injected PD brain lysate into this transgenic mice, and we see the same kind of pathology as the synthetic material, as well as the lysate from symptomatic transgenic mice. So what have we learned from the set of experiments? And what we showed is that misfolded alpha-synuclein alone and can convert endogenous alpha-synuclein in vivo. And the pathological alpha-synuclein species propagate and amplified in the CNS. And this transmission seemed to follow a reproducible pattern and following its neuroanatomical path. And accelerated aggregation strongly correlates with earlier onset of neurodegenerative disease phenotype, suggesting that the pathology is responsible for this early death. So it's great that we can do it in the transgenic mice, and the mice themselves make pathology, although not as robustly as as what I just showed you in the transgenic mice injected with fibrils. But what we want to do to see whether or not we can show the same phenomenon in non-transgenic mice. And after all, we were successful in getting pathology in neurons generated from non-transgenic mice. And so, another reason to really conduct this experiment is that that some of you may know that the current animal models of Parkinson's disease are not perfect. And so there are really two major pathology in Parkinson's disease. One is the loss of dopaminergic cells, and the other is the formation of Lewy bodies. And it has not been possible to link these two pathology together because You can generate mice with dopaminergic neuron loss by lesioning the nigra with toxin like MPDP and 6-hydroxydopamine. But these mice don't develop Lewy bodies, and people have tried for many years. And also many people, including ourselves, obviously, have made mice that overexpress alpha-synuclein and show Lewy body formation, but none of these mice, and nobody's mice, actually develop, although they develop pathology, but they do not lead to dopaminergic cell loss. So we thought, well, maybe we can link the two together and demonstrate the formation of Lewy body and dopaminergic neuron loss in these non-transgenic mice injected with alpha-synuclein fibers. So we basically, I showed you that the transgenic might lead to death. And what we want to do is that if we inject into non-transgenic mice, we're not so bold to suggest that it would lead to death, but perhaps it would lead to phenotype, okay, and motoric phenotype. So what we did was that we injected these normal mice, control mice, and non-transgenic mice with mouse alpha-synuclein fibers. And we, again, took a lesson from the prion field, and that there may be species barrier and in this transmission phenomenon. So we used mouse alpha-synuclein instead of human. And we injected in the dorsal striatum, and because we know that dorsal striatum connect directly to substantial nigra pars compacta. And amazingly, after 30 days, we saw pathology. And you see these, what we call Lewy neurites and in the site of injection in the striatum, and also in the cortex. And surprisingly, some of the olfactory neurons are labeled with phosphosynuclein as well. But by 6 months, and you can see that the striatum definitely show Lewy body formation, and the pathology found in the amygdala, in the frontal cortex, and also in the contralateral cortex as well. And the nigra too, and have pathology. This is the pars compacta, and the green is tyrosine hydroxylase, and you can see that the red is the phosphosynuclein. You can see accumulation of a phosphosynuclein in tyrosine hydroxylase positive cells. And so the pathology spread over time, and the spreading and seemed to follow the anatomical pathway. And you can see 30 days, and we injected right here in the dorsal striatum. And the pathology is in the cortex and in the amygdala, but you can see an over time increase in the amount of pathology in these mice. And pathology look very similar to the human pathology in the sense that they're misfolded and they're ubiquinated, and like the human Lewy body, they recruit an HSP90, and also they're thioflavin positive. And formation of the Lewy body in the nigra lead to progressive dopaminergic neuron loss. And conveniently, we can use the contralateral substantial nigra as a control because they don't really talk to each other. You can see here at 1 month, the TH cells are pretty normal, but by 3 and 6 months, you can see about 50% reduction of the dopaminergic cells. And so the loss of dopaminergic cell lead to a loss of striatal dopamine innervation. And so you can see a reduction of dopamine, and at 180 days by about 50%, and also the metabolites as well. And they do have a coordination impairment in the rotor rod and the wire hang, both of these motor assays that you can, behavioral assays, you can see that by 6 months particularly, that the mice don't behave very well, but they don't have any cognitive impairment at this point. So what have we learned from these studies? And what I've shown you is that mouse alpha-synuclein preformed fibers initiate efficient conversion of endogenous alpha-synuclein protein into Lewy bodies. And these pathological sequence of events occur readily in healthy and wild-type mice. And that fiber-seeded pathology expands and progressively expands within the neuroanatomical connectomes. And so alpha-synuclein inclusion drives the selective loss of substantial nigra pars compacta dopaminergic neurons, resulting in behavior impairment reminiscent of motor deficit in human Parkinson's disease. And so what are the implications of what I have just shown you? So obviously, the original title of my talk is to claim that this is the common mechanism for all neurodegenerative diseases. And so this is really a question we want to answer. And there are already some evidence in the field and showing this type of spreading for both A beta and for tau, and some of the tau work we've done, and I don't have time to talk to you about. So the other thing is that going back to the clinical, to the patients, and what we want to know is that this is spreading of misfolded disease pathology correlate with human neurodegenerative diseases. And this is something that definitely need to be addressed. And then the third point is that, these alpha-synuclein pathology, and they're found in neurons, but they're also in oligodendrocytes and other cell type, and particularly tau is in astrocyte as well. So, and so they also have, the diseases have different clinical phenotype. So why do they have this diverse clinical phenotype and found in so many different cell types? Do they represent strains? You know that in prion disease, there are strains, right? So does this represent strains that can give rise to the different disease phenotypes and different pattern of spread, and also the influence of other pathology in the brain? And as you know, that Alzheimer's brain also have synuclein pathology and TDP-43 inclusions as well. And this novel cell and model of sporadic neurodegenerative disease and provide new opportunity to study mechanism of disease pathogenesis already. And we've collaborated with some other people that have been able to show the same type of paradigm, the spreading that I'm showing you in rats and rat neurons, and also in intact rats. So we inject the fibrils into rats, and they actually transmit and do the same thing as I'm showing you here. So there's a lot of things that we need to do. And so in conclusion, I want to acknowledge people in the lab that have done all the work. And Kevin Luke did the majority of the in vivo study, and Laura Bobuccelli-Dali led the cell culture studies, and all the other people contribute to the study. And our colleague and the School of Engineering help us with the functional study, and the studies are done in collaboration with John Trojanowski, and this is our funding source. I thank you for your attention.
Thank you, Virginia, for a very exciting talk. Yichun?
Okay, I have two questions. First of all, is the spread also occur in, whether you have tested whether spread occurs in synuclein knockout mice?
No, it's not.
It's not, so it's dependent. So that's a nice proof.
Yeah, yeah.
Second is a little more about the mechanism of spread.
Mm-hmm.
You imply that it's anterograde from your cultured cell. Is that the case in?
Right. In cell culture, it goes both direction, both antegrade and retrograde. Yeah.
How about in vivo? Do you know from the spread pattern whether it's mostly anterograde, retrograde, or both, and whether neuronal activity facilitate or decrease?
That's what we want you guys to do. I think that it's open really, opportunities and for the electrophysiologists and people in the audience to really do that. I mean, it is very simple to do, and so I think that it probably is activity dependent, but we don't have any data.
Yes, a quick question. I'm wondering whether have you ever checked about the mitochondrial clearance of the neuron, which has been transferred by the alpha-synuclein?
Right. The mitochondria actually are fine, and we haven't really looked at them in great detail in the neurons that are degenerating, but the transport of mitochondria in some of our new experiments are not compromised at all. Fast transport is not compromised by the pathology.
Okay, thank you.
Yeah, I had a question about the cellular compartment where this interaction might take place between the pathological synuclein and the endogenous synuclein. So I was wondering if you could comment a little bit about where the native synuclein normally sits, and given that the pathological synuclein would need to be taken up by an endosome or something, where this intersection you think happens or
That's
will
right.
happen.
So this is a very important point in the sense that the way these little bits of fibers are taken up sort of define conventional cell biology in the sense that, that we think that endosome takes it up and then, but then this thing is so chunky. So maybe it just, caused the endosome it may leak out. Or they may be totally complete different new mechanism of endocytosis. So again, it creates new opportunity to study this phenomenon. How does it get into the cytosol, and it's not supposed to do that, but they do. So a lot to do.
Okay, I have a question here.
Yes. Okay.
It seemed that PFF alpha-synuclein is necessary for the Y-type alpha-synuclein propagation in the CNS. I'm wondering whether this PFF is also necessary for the host to graft propagation.
It's necessary to graft?
Host to graft propagation.
To propagate? No. So you have, basically the way it works is that you have a little bit of seed, whether or not there are ligaments or little bits of fibrous. And so it serves as a template to corrupt the endogenous protein, to adopt the bad conformation, and then, propagate. And so we don't know how it's being released. The mechanism of release is unclear. And the mechanism of uptake is unclear. And how it spread inside is unclear. And all I can say is that it probably is the bad conformation that is corrupting the endogenous good conformation. So.
If you treat the stem cell with the preformed fibril alpha-synuclein, will the stem cell develop to Lewy body-like structure or something else?
I think that because it depends and critically on the concentration of alpha-synuclein in neurons. And we know that, the alpha-synuclein are not really expressed until synapses are formed. So they're really abundant in the presynaptic terminal. And so, for example, human neuron derived from iPS cells, and they really have not been mature enough to really allow this phenomenon to occur. Other people have done those experiments, but I think that you need a high concentration of synuclein in mature neurons.
Thank you.
Question in the back.
Do you think estroglia will increase the spreading of alpha-synuclein in your in vivo model after you inject alpha-synuclein? Do you see any inflammation?
There's really very little inflammation. Yeah, not much inflammation to speak of. Yeah.
Tom?
Virginia, very nice. I have a question that regards the size of the fibrils, both for the transfer and for the transmission. In view of all this discussion of oligomers versus elasis versus amyloid, at least for the seeding, you could potentially look at different sized fibers, right? And also potentially for the transmission. Do you have any idea of what's the dependence of the...
Right. So we're in the process of doing those experiments. But what you need to do is that you need to fractionate, right, your preparation to really identify the smallest species that would do this. And what I can tell you is that there's soluble material that can do it. How large they are, we don't know. Whether or not they have the same specific activity as those smaller aggregated species, we don't know. And but those need to be done and to try to find out the species that are most active. Yes.
Question in the back.
On the tissue culture method that you used, the cells were top to top on the same membrane, or are the lower cell on the bottom and the top cell on the membrane? I didn't get that.
And
the two-chamber method.
Oh yes, yeah.
The cells, they have a neuron on the lower chamber, neuron on the top chamber.
Right.
Are they attached on the same membrane, or one attached to the?
They're in this device that you buy from this company, and then basically they're sealed such that only the axons, the processes, can go through this narrow groove from one chamber to another.
So the size of the pore, how much? How big is the pore size?
I don't know exactly how large, but you can actually, and there's a study published recently to show that we show that the pathology can go across from one compartment to another. But what these people showed is that the seeds actually can be taken up and transported along the axon. So,
So in that case, have you looked at the possibility of something, the inclusion are always cytoplasmic, never have a double membrane around. The inclusion body is always cytoplasmic. There is no membrane.
You know, you don't really see the inclusion.
Okay, so have you looked for the lipid raft
yes.
possibility
Right.
that this inclusion can be transmitted from one cell to the other, because it looks like a basolateral on the pictures.
Right. Yeah.
Let's continue that discussion. Last question.
Okay, I have a question. Since the fibril is such a powerful seed, do you have any idea that you may be able to block the seed spreading?
That would be great if we can do that.
But what's the mechanism under the fibrillation? I mean, because you did that in vitro, and then once it's you put it in the cell, so it kind of spread, right? And nuclear. So what is the biochemical process really involved?
So according to Stan Prusner, is that you have a bad conformation that corrupt the endogenous protein to adopt the bad conformation, and then so more and more, and they spread. So it's a very simple, you know,
and
idea.
Once the seed is there, how do we block that? More proteins coming up.
I think blocking the protein-protein interaction may be hard, but still, I think there are people in pharmaceutical company may want to try, but that's traditionally have been difficult to do. But maybe you can block uptake, and you can, you know, block release, or you can, you know. Yeah.
Okay. Well, let's thank Virginia again. So there was a cell phone that was picked up. It's of the brand QPAC. And the cell phone is now at the hotel concierge desk. And if you have lost it, please go claim it, okay? So we now take a 15-minute coffee break.